We are going live now. All right. So, Miss Emmanuel, can you hear me? Hello, Emmanuel.
Yes, sir.
All right. That's great.
Yes, sir.
So, good evening, everyone.
Yeah, good afternoon, sir.
Thank you. So,
Yes,
very
sir.
quickly, we'll be looking at one of our major webinars, which we do every month. And today in the Anatomy Trial Foundation, we are trying to look at bioinformatics. So today we'll have the person of Emmanuel that will take us through bioinformatics. He will definitely introduce himself better, and then thereafter we'll be going forward to looking at KOBO TOOLBOX. All right, so Mr. Emmanuel, you can go ahead.
All right, sir. Good afternoon, everyone. Good afternoon. So, it's actually a great privilege to be here this afternoon, and I want to use this moment to appreciate our beloved president, the president of Mr. Matthew. Thank you so much for this privilege, and also want to use this to appreciate all anatomy triad family. Everyone is striving to be better. So it's good to have everyone here this afternoon, and I hope
Please,
that
Mr. Emmanuel, you can
yes, sir.
unlock your camera is off.
Okay,
All right, we can see you now.
okay. Thank you so much. I'm actually in the lab in our animal house. So, all right. So, good afternoon, everyone. So we have quite a number of things to do together this afternoon, but I will just move on and just try my best to cover as much as possible because bioinformatics is actually a wide course. All right, so I'm Emmanuel, by the way, and currently I work as research assistant at the Eagles Research Lab here in Ladoke, Akintola University of Technology, Ogbomoso. All right, so I'll be putting us through bioinformatics, which is not actually a new field, but something interesting, a field that is actually comprises everyone, irrespective of your cost of study. And I hope you find this interesting. Okay, so what is bioinformatics? So when we talk about bioinformatics, we have to actually define what it means. All right, so bioinformatics is an interdisciplinary research area that interface between computer science and biological science. So it is responsible for storing, analyzing, interpreting big data generated by life sciences studies or acquired in clinical sciences. Now, what bioinformatics actually help us to is to help us reduce time. There are some things that we can actually achieve at faster rates. So, and instead of we going to the lab, experimenting, trying some things, we can actually use a computer with the help of AI algorithm. We can use it to extract some information that can save stress and also give us some insights about some things. And we can use it to model. We can use it to predict what is actually what's the result going to be like, what's the outcome of a particular um, research, right? So it is actually very important that everyone should have the idea of this because bioinformatics also it serves as a link between wet lab and a desk work. Well, what I mean by desk work, that is using of the use of AI to analyze some things. So we can start some things using bioinformatics knowledge. Then when we are able to, okay, we are able to get the prototype of the thing, then we just move on straight up to wet lab and then carry out the was prototype and boom, we have our result. So bioinformatics cuts across every clinical or biological science area. So it is very important. It is the new hot cake right now. And as a young person in finding is our place in research, it is important that we can actually dive into bioinformatics. So we have different application of bioinformatics. We can apply it in plants, viruses, anything clinical setting, anything that can improve life, right? So that is bioinformatics for us. So you can say it integrates computer science knowledge, biology students, anatomy students, physiology. It serves as a background, it serves as a meeting point for all these uh, courses, even biochemistry, right? So, but as there are quite a number of advantages to bioinformatics, also there are some disadvantages in which we are going to look into as we move forward, right? So, bioinformatics is actually limited to sequence, structural, and functional analysis of genes and genomes and their corresponding products. So I'm going to take my time to explain this. Now, when we talk about bioinformatics, we are trying to go to the cellular level. Yeah, we are trying to analyze some things uh, at the cellular level, right? So for instance, um, for anything that any disease, so before we can let me take our body for example. Now, before a body can collapse, maybe someone is sick, it shows that there has been some things that has gone wrong at the cellular level. So for anything that we see, any disease, any effect that happens to human being that we can, like for instance, cancer. Now, so cancer don't just show up. It started at the what? At the molecular, the cellular level. So bioinformatics actually deals with that. It is limited to sequence, structural, and functional analysis of gene. So when we talk about gene, we are talking about DNA, RNA. Now, for every gene, most genes code for proteins, right? So by the time we move on, we are going to see more about the importance of bioinformatics. All right, so but there is actually a difference between bioinformatics and computational biology, right? So computational biology is actually more confined to the theoretical development of algorithm used for bioinformatics. Now, um, computational biology deals with we developing an AI and algorithm that can actually help us to 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 make some findings apart on a particular gene or anything related to genes or anything related to the cell. So computational biology is actually different from bioinformatics. So some people think they are the same. They are not actually the same. So computational biology deals with the development of the algorithm that we use in what bioinformatics. Okay, so we move on to this table. I don't know if we can actually see this clearly. But I'll try to
I
explain
think
this now.
you can go ahead and presenting it. Like
Okay, okay,
now, what I mean
okay,
is by clicking on your presentation from the Microsoft PowerPoint button, as if you want to
okay,
project it in a wider screen.
okay. Let me
Yeah.
see if that works. How about I've done that? I don't know if it's actually reflecting from your end.
Has it been able to come out from your end?
Yes, it's full screen now. Yeah.
All right.
Okay. How about now?
Just go ahead.
Okay. Let me represent my entire screen then so that you'll be able to have the full screen. Coming. Can we see the full screen now?
It has not come up.
Okay. Okay. Working on that. Probably network. We're showing that I've started my screen sharing. Hello. Mm-mm. I don't know what's wrong with the network. Well, it's sharing from my own hand, yeah. So I don't know why it's not actually coming up. Mr. Master, can you see me? Okay, I think it's up now. Okay. All right. So sorry for the breaking transmission. All right. So this is actually the core application of bioinformatics. So we have the structure analysis, sequence analysis, and functional analysis of bioinformatics. So now, don't forget, actually the bioinformatics is actually is based on the cellular level. So we want to do some things at the cellular level. So some of the things that we can actually do at the cellular level is to predict the nucleic acid structure prediction, the protein structure prediction, the protein structure classification, and the protein structure comparison. That is at the structure analysis level. Now, I will try my best to break this down. Now, when we talk about this structure analysis, now for a particular human being, now we have what we call genome. Now, genome is the complete set of genes that we have in a particular person, right? So we have something, we also have quantity. So those are just from a few best pair of nucleotide, right? So we can actually predict the acidic nucleic acid prediction of a particular body. And also I said mostly most genes produce protein. which are essential building block for the human being. So for every gene, they code for in the DNA, the code for a gene, and the gene will code for a protein. Now, this protein is what is now functional in our body. So if there is any if there is a tamper with the structure of a particular protein in the body, that is when we have some defects, we have some diseases, we have some anomalies, we have a lot of effects at at the macro level, that is at the gross level. So the protein structure, we can actually use bioinformatics to predict some protein structure. Now, for example, bioinformatics is actually used also to produce to predict the 3D structure of some proteins, which is very essential. Now, for example, now we know that Alzheimer's disease now is actually it is related to amyloid beta, right? Now, this amyloid beta is a protein. So we can use bioinformatics to also predict what gene can actually produce this particular protein. And that is why when we study neurodegenerative disease, you'll discover that there are some genes that are being targeted if we are going to diagnose some people as maybe a potential Parkinson patient or Alzheimer's patient, etc. So there are some genes that we can target for this analysis. That is where the structure analysis comes in. So we have the the nucleotide structure prediction. We also have the protein structure prediction. Now, don't forget, from the nucleotide, we can predict the protein. So we are going to look into some of the databases we use for this particular structure analysis. Now, we also have sequence analysis. This is actually very interesting. I'm going to share with us some of the products, some of the research we actually carried out sometimes last year concerning this sequence analysis. So when we get there, we are going to get more about this. So this sequence analysis means that we are trying to compare two or what's it called sequences to, in fact, more than two sequences. We can, and that one of the database we use for that is blast. All right, so we get to the bridge soon. So genome comparison, what are the things, what are the difference between a particular genome and another particular genome? So we have the motif discovery, we have the sequence database searching, we also have what we call sequence alignment. Now, all these are actually essential because one of the applications of bioinformatics is we can actually use it in designing a diagnostic kit. Yes, use bioinformatics knowledge to design diagnostic kit, also in vaccine design. Remember, all these are actually connected to the cellular level of genes, right? So we have the function analysis. Yes, good. Because this function analysis actually predicts the metabolic pathway in which a particular gene, a particular protein can actually interact. Now, like I said, I'm going to share one of the projects we carried out on with this function analysis. Now, bioinformatics, thank God for the databases that are available now. There's this particular research, the one we carried out, right? We were trying to check why some organisms actually have some AMR, that's antimicrobial um, resistance, right? So there are some strains, there are some genes that allow some strains of organisms not to be affected by some antibiotics. So this is where the functional analysis comes in. So we have some database that have helped us to bring some of these genes and their functions. So this database gave us the metabolic pathway in modeling. So, like I said, this is also applicable when we want to design a vaccine design or diagnostic kit. So the next one is database. Now, what is database? Now, databases are store of biological and genomic data that are built to store, organize, and provide access to a wide range of biological information. Right. So there are one of the reason why we carry out this research I'm talking about, in which I'm going to share the full details soon, is that we want to compare a particular organism, the strain, the serotype. We want to compare them, what makes them differ from one location to another location, right? So that anybody who wants to develop a vaccine or want to develop any diagnostic kit for this particular disease or for this particular organism can actually have access. So when we are done with this, we will upload it to a database. So a database is like a store that, okay, for example, there's already a primer to target a parking gene, right? So how do we get the primer? It is due to some of the records that we have on database. So we have different types of database, which is very important in bioinformatics. So they are just like a storehouse where genomic, don't forget we are laying more emphasis on the cellular level. So, right, so biological and genomic data that are built to store, organize, and provide access to a wide range of biological information. Right. So we have different types of biological databases actually. So they are categorized based on their content. So we have theory, we have the primary database, we have the secondary database, and we have the specialized database. So when we talk about the primary database, secondary database. So the primary database is the raw form of some data that has not been processed, that has not been worked on. Just maybe, for example, Drosophila melanogaster. I can actually go to a particular website in which I'm going to share um, the slide soon. A particular website that can actually give us the whole genome. Genome is the complete set of genes of a particular organism. So the complete genome of Drosophila is actually available. So this database gives us access to this particular genome. So whenever we want to um, maybe develop a primer to target a particular gene, we know what to target. We know the type of primer we are going to develop so that we can target that particular gene. That is another aspect entirely. But this database, permanent database, actually give us the raw file that has not been worked on, that has not been modified, right? So the secondary database is based on some of things that we have modified. For example, you can actually download the nucleotide, the DNA. When I'm talking about nucleotide, so the nucleotide of a particular organism, then you can convert it, take it to a particular another database to change it from nucleotide to protein. So in that format, that means there have been modification from nucleotide to protein. So now this type of database is called the secondary database that has been modified a little, right? Maybe from nucleotide to protein and from protein down to the structure, right? So another form of database that we have is specialized database. So this specialized database are actually databases that are unique for a particular work. Now, for example, we have HIV database, we have Drosophila database, we have C. elegans database. Instead of we going up looking for a particular data in another database, since we know that okay, Drosophila database are mostly operated at this database, that is a Firefly database, we can just go straight to that database, and everything that we can find in that database is nothing but Drosophila related. So that is what we call specialized database. We also have some database like cancer database, right, and the likes. All right, so these are the list of databases that we have on the retrieval system, and we have the summary of their content and the website, right? So one of the most common database that we use is NCBI. Yes, so this NCBI is just like a junction where other database are also incorporated. For example, from NCBI, you can actually dive from in NCBI into PubMed, right? So these are actually some of the things. Maybe I'll find a way to send this to the group or to attach it after this section. So I want to move fast. So there are some database that we have now. For example, SCBI, which is National Center for Biotechnology Information. So these actually deals with nucleotide sequence database. In fact, there are lots of database that are also incorporated into this particular database. It is a why it sounds like a father to some database. Let me just put it that way. So, like I said, there are some specialized database. No, so NCBI is not actually a specialized database. Why? Because you can from NCBI you can get nucleotide, you can get protein, of any particular organism that you're looking for, right? So for this, we have MGI, which is mouse genome informatics. So this is actually a specialized database which is related to genomics of mouse. Now, the genome of human is not readily available, right? So then when you say genome, that's the complete set of genes, right? So it is not actually available like that. So what we can actually have for human being is the content just from there are some things that we need to learn in bioinformatics. Bioinformatics is not actually a field that we can just dive in without adequate knowledge, right? So we have to have some adequate knowledge, and one of the adequate knowledge we have to have is on cell. Yes, cell, DNA, RNA, the messengers, everything related to cell. It's actually going to help us, especially the central dogma. Also, we can read on it because we have to, there are some limitations to bioinformatics. So whenever we are trying to work on this work on bioinformatics, we have to be sure that what we are doing is correct. Yeah, so that we won't waste our time. So another database that we have is ENA, which is European Nucleotide Archive. It is also a good database to retrieve some things. So we have UniProt. This is Universal Protein Resources, protein sequence and annotation database. Now there's something we call annotation, that is just to mark something, right? To give a particular sequence a name, to note it when we have worked on it, right? So we have also Maze GDB. Now, you might be surprised at what is Maze actually doing here, right? So bioinformatics is also related it is related to plants. So we have the plants, we have the organism, we have different aspects of biochemical sciences that we can actually relate bioinformatics to, provided it is a living thing. Yeah, provided it is a living thing, provided it has cells, provided it has genes, DNA. All right, so we have some bioinformatics web servers, which are platforms that gives us the access to analyze, and they are actually user friendly. We can navigate our work ourselves through it. So we have some of them like StringDB. We have this for the is it provide information and tools for analyzing of protein to protein interaction, right? So remember that I mentioned earlier that genes code for proteins, and these proteins are actually what we the information which the body use to carry out their function, right? So protein to protein, how does this the proteins that are being produced by this what's it called? The genes, how do they interact with one another? So that is it's also used to studying the functional interaction between proteins, right? So we have also a very good website here, which is Galaxy. So Galaxy helps us in visualization. It's help us to um it give us some access to different analysis too, right? So we are going to explore that in short while, right? So I'm just trying to give us some importance of bioinformatics, the web servers and their use. Okay, we also have this particular server and their uses. And this particular one is called UCSC Genome Browser. So this one is based on visualization and exploration of genomic data, right? Everything is still talking about the cellular level. So as an anatomy student, we have to build interest in cellular and molecular biology, yes, for us to be able to relate well in this particular um field, right? So I'm so privileged to actually be introduced to bioinformatics during my CWS program, and when I was in 400 level. So this actually helped me to develop interest in bioinformatics. So I have to do quite a lot of studies so that I won't be left out, because biochemistry students could relate well, and to some extent physiology students can also relate, but biology students find it quite easier, and biotechnology students. But it's not actually hard. All we have to do is to develop a particular to develop interest in it. All right. So this particular one is one of the things that we were able to achieve doing our own project, our research work. So I'm going to take my time to just put us through this so that we can be able to get some things done. Right. So let me just tell you a brief what's it called, story about this particular project that we carried out. So then we carried out a particular project on E. coli, that's an organism, a bacterial organism. So this E. coli have different classes. So we have a particular one that we focused on was the enteropathogenic E. coli. So this enteropathogenic E. coli is the organism that actually causes diarrhea in infants. So between age 0 to 9, thereabout. So this EPEC causes diarrhea in infants in that age bracket, right? So what we were trying to do is, okay, let us carry out a research that can actually help people who want to develop a vaccine or drug, yes, solution to this particular EPEC, that is enteropathogenic E. coli. So what we did was, there are some drugs that can actually work on white people, and yet when a black person take it, it won't have much effect, right? Maybe it might not even really work on black people. So we were trying to pick out genes, some genes that are common in different continents. So for Africa, for example, now we picked 5 countries in Africa. We picked 5 countries in Asia continent. We picked I think 3 in Europe, right? So, but one of the challenges that we had is that Nigeria does not really have some sequence on EPC on this particular bacteria. So we couldn't find any sequence on this particular organism in Nigeria. So we had no choice than to go for other African countries like Mali, Gambia, yes, Mali, Gambia, Kenya, yes. So because at that particular in that particular country, we discovered that the rate of diarrhea in infants is actually high. So we actually target them. So we were able to download some sequences. And when we are talking about sequence, we are trying to get the genome, yeah, the whole genome sequence. We ensure that we target the whole genome sequence of this particular organism, right? So for us to actually get the whole genome of this particular organism, so I'm going to be sharing my. Okay, I hope you cannot see my screen. So we just do a rough walk of how this actually works. So we were able to get some whole genomes from of this particular organism from other countries that we targeted. Actually, we targeted about 50 countries, but unfortunately, because we couldn't get the whole genome, which is one of the limitation in bioinformatics, there are some data that are not readily available, right? So we are unable to get the whole genome. We only we only got some which are counting. So, but for us to be able to analyze what we want to do, we actually need the whole genome. So this whole genome of this particular organism were downloaded from NCBI. All right. So and one of the thing about this research is that we have to look at the source of this sequence. So I'm going to put us through now how we actually got that. So this is NCBI. Okay, let me just open a particular new tab. So you just see, I just want us to build interest in this particular feed, that is because it's something that is wide and we need more people there, right? So this is the interface to NCBI, right? So for me to able to get some things, so we you can search at this particular part. So now what we make use of in for this particular research was, instead of at this particular point we have all database, so we can streamline this to nucleotide. So let me just at this particular part we can streamline it to nucleotide. What I'm trying to say, I just need the nucleotide for this particular organism. So when you know the name of the organism, you can actually search. You can search. Now we have some things, right? So let me just explain this. Though we have the sequence downloaded Now, for this particular one, now this is EPEC, this is um, enterogenic, enteropathogenic E. coli. So for the, con- I'm actually interested in this particular bacteria. So I will have to search and be careful. There's okay, good. Look at this particular one now. We have 1,446 base pair. Now, when we say base pair, those are the two terms that we need to understand in bioinformatics. So when we talk about base pair, DNA is a double-stranded structure, right? So we have to complement, which have to complement one another. For example, in DNA, we have adenine pairing with thymine, that is HCGC, right? So for this base pair, that means we have 1,446 base pair. Now, the limitation to this is it is not actually the whole genome, it is a shotgun sequence. That means this particular file, this particular sequence only um was able to target a particular length of the whole genome. Now, I just want to walk us through how we were able to get some things, right? So, keywords is whole genome sequence. Now, for some of this, the source, one of the things that gave us work that makes it more tedious is that we have to come down to this particular level and search for the source, because these are the details of this particular strain before we download it. So we have to search for the source. The source that we are actually concerned with is physics. So because EPEC actually is transmitted through physics, right? So when there's a particular place where the sanitation level is very low, so when we just in a location where they defecate and throw the poo outside, uh, just in the public space, they are more prone to have what's they call EPEC, that is this bacteria which can actually cause diarrhea in infant. Now, I don't know if this can. I don't know how to zoom it. Okay, let me try my best. I think I can zoom. Just want to show us how we have to be. You have to have. Okay, I think this is more clear. I've zoomed it now. All right, now this isolation source. It is what the one I highlighted. It is isolation source is two, and that is what we actually want to make use of. Like the samples is actually gotten from these two, from feces. So what's the host? The host is Homo sapiens, human being. So good. When we have something like this, we all we have to do. This is a good catch. Now we can just come here to download. So this is actually for the protein ID. Now, don't forget what we had was we had nucleotide, not protein. So it is the nucleotide I want to work with. So one of the formats in which we can download this particular sequence is in FASTA. Some people can call it FASTA. So FASTA. So when I click on FASTA, right, it changes it. Now this is the sequence for this particular strain, for this particular organism, right? But don't forget, I'm just using this for example. This is not the whole genome of this particular organism, but it's because of our time, I just have to use this as a sample, right? This is it. We have this, you can see GAT and everything. Those are the nucleotide of this particular organism for this particular strain, right? So we have to download it. So One of the ways to actually download it in faster format, you can have this. This is faster format. We have also some formats, but faster format is what most of these database actually likes to use for to analyze, right? So when I click on send to, I come to file, then it said which format do I want, which is past A. Yeah, I don't need other stuff. Genbag is also something entirely different. So I've downloaded it. So I've downloaded the sequence here. So that was how we were able to download some sequences majorly for the whole genome sequence. I have them on my system, but because of our time, I can't really search for it. So that is how to search. You can see the tedious work, getting the source, getting the host, so that we won't um, download the wrong file. Now, when you download the wrong file, when you are not careful, maybe you want to do one you are not patient, you just want to get the work done. What if you could choose the host to be the host to should be a monkey? Now you mistakenly download a monkey genome and you went on to analyze. You are going to get a wrong you are going to get a wrong result of which you cannot really detect. So that is why it is very good to pay every little details is actually what important. So after downloading the nucleotide, um, what's it called? The nucleotide for all this whole genome sequence of this particular EPEC, we went on to another database, which is BVBRC. So BVBRC is actually a database, a specialized database for bacteria and viruses. Now you can see the full meaning here. Now at this particular point, all we have to do is now for that particular sequence that we downloaded, this database actually gave us the opportunity to um get the valence factor of each sequence. We have about 50 sequences that we downloaded. So we have to check their valence factors. So that means the genes that make them to be valent. And thank God for this particular database. It helps us to get these genes that makes this particular organism toxic, pathogenic in nature. So we make use of it. Time will not permit, so we can what we're actually working on is bacteria, right? So you can okay. So tools and services. When we click on tools and services, we have quite a number of things that we can make use of here. So, but one thing I need to say also is that it depends on what you are trying to carry out, the type of project you are trying to carry out will determine the type of database you are going to use. So, luckily for me, we have two institutions here in Ogbomoso that actually works on bioinformatics. One of Ellis Bowden Institute, which is actually highly recommendable, and also a Genoma Hub here in Ogbomoso. So, well, I've been privileged to be in one, and I also have the privilege to have colleagues who also pass through Genoma. So sometimes when we share things together, then we discover that mostly we use different database to get our results. So this actually gives us the 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 valence factor of this particular organism, right? So we have specialty genes. Okay, this is where you come, then you upload your want to put this. All right, so let me say EPC. Trying to work on some things, but all right. So one of the disadvantage to this particular stuff is that sometimes the database can have can how will I put it? Yeah, there might be a lot of workload on the database, and sometimes might not actually process what you try to input in, right? So that is for this BVRC. So it helps us to get the balance factor. In fact, we will get the exact format of this particular organism. Yeah, we get the particular exact file. So from this particular part, I can download as table, I can download as CSV or Excel, which is very important. And trying to navigate through something here, but without wasting much of our time, let me just move on. So that is what we can actually use BVBRC. It is actually used for um it is actually used for bacteria and virus analysis and everything. So the next thing we did was okay, good. You can see I've searched for him, and it's just bringing out the stuff. So I can filter. So one of the things that we did was we filtered, right? So remember we chose some particular country. So when we get look at these are um some particular countries that we have. So I think Mali. Okay, wow, Nigeria is not there now. So as at then when we worked, there was we don't have Nigeria there actually. So we can come here, start to filter, we say complete. When we say complete, we have some things that I would have done. We choose Gambia. So since Gambia, then we have this particular strain. Now, when we download this, we can download this and have them in our exact format. So when we download them, we can actually okay, I can download now. Let me see. Exact. So it's downloaded. So one of the disadvantages with bioinformatics is it makes use of a lot of data and power supply. So let's see how this actually works. Sorry for taking much of our time. I think I'm far behind time, but we're just trying to walk you around this particular. Okay, so trying to open the file I just downloaded. So this database actually, you can see how we have been able to filter it, narrow it down to what we actually need. So that was what we did to get some the sequence. Let me, I don't know what's wrong. Okay, but maybe when it's open. So I've downloaded it. Okay, I think it's opening. Yeah, and your system also determines how you are going to enjoy this particular um bioinformatics, right? So we've downloaded two things now, the sequence in faster format and also the genome from this particular, sorry, my system's almost lagging. Sometimes in fact, one of the limitation in Nigeria is that when you want to work in with bioinformatics, you have to have a constant power supply. If you can see me very well now, I'm actually in our animal house because of constant electricity. Okay, so while we wait for that to actually load, let me just move back to this. So what this database actually helps us with, it gives us the genes. Now from this particular genes, when we get the genes, when we when we're done with this particular BVRC um, database, we had about more than 5,000 genes altogether from the 50 sequences that we downloaded from different countries across all um, continents. Now when we were able to get these genes, so we check for their antimicrobial resistance of some things. So we use CAD. This is also a database that can actually predict some of these. It's a database that can predict the genes present, right, and their function and the antibiotics that they can actually resist. Now, for example, I've searched one here for the course of this class. So let me copy. So a card, just search for card database. So we have this, and when you press enter, so we have it will load, right? I don't know what. Okay, I think I'm working faster than my system, so I'm just okay. Let me just use this. So when I've actually searched for this particular gene, now this gene were actually okay. Good, we have this. This is what we actually downloaded from the BVRC. So it gives us the information, the code number, the type of kingdom, the class, the order, the family, the species. So it gave us quite a number of information. So for this one now, you can see when we move on, we have quite a number of information that we can work with. So this gives us the more information about this particular stream that we are looking for and some of the things we can actually work on. So um because of what I actually clicked on. Okay, so let's move on to this. When we get the gene, we're able to start from this card. Now for this particular one, it gave us the card short name, which is this is a particular gene. Now this is the function of this gene. So in the presence of antibiotic stress, E. coli overexpresses the global activator protein resist. So what are the antibiotic drugs that they can actually resist? We can have quite a number of them. So what this actually project aim at is what are the genes that are common to all these sequences that we downloaded, so that whenever we want to, when anybody wants to develop a vaccine or a drug against this particular disease, which is diarrhea, so we can actually target something that will make the drug very potent, that when they use it here in Nigeria, it will work, and also in Asia, it will have the same effect. Not that because of the strain that are different will actually counter the effects of this particular drug or vaccine. So we can see quite a number of them that the drug class that this particular gene, presence of this gene can actually make the organism be resistant to these antibiotics. So it's not about increasing the dose of the antibiotics now, but it's the function, right? So we're able to get this, and that is for the card. And the another thing that we did was checking for the strain and the pathogen finder. So we'll be able to know if this thing is actually pathogenic to human being or not. So we use this particular word, Center for Genomic Epidemiology. Is actually quite a lot word. So we can upload the sequence that we downloaded, the sequence here this time around. To download, you input it. So I have some of them open, then upload. So this will actually give you, it's loading, right? So this is 5.1 MB. So it's, you can actually input your email so that you can leave this particular one, or you wait. The page will actually update itself. Now that means you have to be connected to the internet. So that is for this particular one, gives us the pathogenicity, the stereotype of all this. So it's something that we can't just learn, but we have to read around it. So I'm just using this moment to teach us how in bioinformatics can be used to develop drug or vaccine, right? So by the time we get all this information, it will be very much easier for anybody that is going to do the wet lab um, experiment to just target this particular gene and know what to work with, right? So one of the things that we also did, which this is where I'm going to wrap everything up, after getting the pathogenicity, after getting some things. So this is actually the document of our write-up. So we're able to, okay, all right. So this is the document of the work. So we using this particular database, we were able to um, get the genes that cut across all these sequences. So, but we didn't get. I told you we had about 5,000 plus, close to 10,000, if I'm not mistaken, of genes that we have. So how do we know which genes are actually common to everything? So we can't do that by ourselves. So we have to develop an algorithm by using Python to compare and contrast every gene from across all the continents and the sequences to know um which of these genes are actually common to Nigeria, to Africa, to Asia, and to Europe. Where we were able to get quite a number, but now this time around it is much what reduced, right? So and we're able to get the protein that they actually code for and what makes them pathogen. Now this particular structure is what we call the phylogenetic tree. We use a particular database also, which is ETOL, ITOL, right? ITOLY. So we're able to use the ITOL to get this. Now, this is the number of the sequences that we downloaded in different colors, right? Now we are trying to relate. How are they related? Where is the source? So we discovered that some of these traits are actually common. Maybe the Gambia one is actually related to India. They are connected somehow, just to just to get the traces of how all these things are actually entwined. So I taught Apples how to draw the tree for this particular sequences for the sequences that we downloaded. So that's what we were able to do. So bioinformatics is actually an interesting field that we need to look into, and it doesn't call for laziness. We have to study because every decision, everything that we do, we must have a concrete backing, right? So we have something that I actually want to uh, talk on, which is blast. So that is one of the techniques we use to relate, what's it called, to compare. Sorry, actually being distracted. Yeah. So to compare the similarities and everything. So there's something we call primer also. So this bioinformatics also help us in coming up with primers and the like. So thank you so much for having me. And I want to say this also, that it is very important that we know some limitations to bar. Right. So hello. So some of the limitations. All right. So
Mr. Emmanuel, can you hear me?
yes, sir. I can hear you, sir. Can you hear me, sir?
All right.
Hello, can you hear me, sir?
All right. Are you done with your section?
Yes, sir. Yes, sir. Yes, I'm done, sir. Can you hear me, sir?
All right. So, Emma, can you hear me now?
Yes, sir.
So,
I can hear you, sir. Can you hear me? Hello, can you hear me, sir?
All right. Ima, can you hear me now? Okay, so if you can hear me, I think,
Yes, I can hear
okay,
you.
all right, all right. We're just having issues with network here because it's about to rain over my end. So
Oh,
that is why
okay.
it looks as if I'm just coming on and off. So
All
thank
right.
you very much for your presentation so far. Thank you very
Yes, sir.
much for your presentation. So if you notice very well, Mr. Emmanuel has been very much involved in bioinformatics, and why we from the Anatomy Triad Foundation have decided to come up with an introduction to bioinformatics from Emmanuel is that he is just a recent graduate, and you could see how much he's. So it is not a do or die affair that you remain in anatomy, or you remain in biochemistry, or you remain in physiology. You have to diversify as you keep growing. So just like him currently is into bioinformatics, he can use that, apply the knowledge, and then he will do better in his own area of specialty. So you too, bioinformatics is an evolving area. So we are encouraging people to see need to diversify too. So if you have access to any opportunity in bioinformatics, you can.
Okay. I think his network is actually poor. So let me see. If you're online and you have a question, you can actually type in your question. You can hear me. So you're welcome, sir.
Okay, I believe Emmanuel, you can hear me now.
Yes, sir. Very much, sir.
I'm so sorry for the continuous glitch. So
Uh,
because of that, we are looking forward to, if you have any questions as it concerns bioinformatics, you can
okay.
just type it on the chat box on YouTube and we'll respond to it. So if you have any question on bioinformatics, how to go about going to bioinformatics as an anatomist, physiologist, biochemistry, pharmacology, and whatsoever field you are currently in that is science related, biomedical sciences, you can still need to ask it now as Mr. Emmanuel will still need to respond to you. Though this training was supposed to be two in one, the first section was supposed to be introduction to bioinformatics that will help you to understand what people are doing in bioinformatics. And then secondly would have been KOBO TOOLBOX, one of the current data collection tool that is being used in the field to collect data either in communities or when you are doing a research that has to do with data. But because of time and because of the weather currently here that might end up disrupting our network, we will still need to take this as our first section for bioinformatics for biomedical scientists. And then subsequently, we have to take KOBO toolbox in our next training section. So this is just like an eye-opening to what bioinformatics is all about. So if definitely you want to know more, just like Mr. Emmanuel Adejobi currently knows, you can definitely make it out by reaching out to us via the Anatomy Triad Foundation. You can send us a mail, anatomytriad@gmail.com. We can try to link you up with Emmanuel and you people can see it on how you can strategize to get to know more and how to have the vast knowledge in bioinformatics just like he has currently. So if you have any questions, we are still waiting.
So in the meantime, I want to also appreciate everyone for your support. I want to say special appreciation to our boss, Mr. Matthew, for having me here today. Um, it's a privilege I can never take for granted, and I hope I'll always be there also in time of reckoning. And, performance is actually an interesting field that we need to actually venture into, especially in molecular biology. If you have interest in research, you can actually um, diving into bioinformatics. Even though it is new, you can there's also something we call neuroinformatics also. So all right, thank you, Mr. Sunday Harper. Thank you. I hope the presentation actually um, impart, and I hope I'm looking forward to serving you better also. So if you have any question, and also you can read online also. There's some textbooks I have that I can actually help you. Easy step guide some things you need to know. You have to understand some things we use in bioinformatics. So I'm still I'm much available. If you can hear me, you can type in your questions so I can respond to them.
All right. So I just believe due to all these glitches coming from. Okay, somebody has a. Okay, yes.
Yeah, so
Uh,
sound
Dr.
question.
Sunday Yaba, that is HOD from Kogi State University, currently Prince Abubakar. He's thanking you already. Thank you, sir. All right, so someone has a
Thank you.
question. Juliana.
Okay, sir.
So he's saying, my question is that after getting the DNA sequence, will one come from the sequence predict? Okay, will one come from the sequence predict the disease type and gene responsible for it? Or that's the function of is it specific database?
Specific
So what
database.
the person
Okay,
is, did you get the question?
okay. Yes, sir. Yes, sir. Yes, sir. I got the question. So,
All right.
thank you for that question. Okay, so for this particular question, when you actually download your DNA sequence, your DNA sequence is actually in a simpler form, like what I just showed. The the pair base of this sequence. So for a nucleotide sequence, all you will see in the sequence when you download it in a fast A format, what you will see is how these bases are being paired, like adenine pairing with thymine, then guanine pairing with cytosine, right? So that is just what the sequence is all about. Now it is until you submit it to another specific database. That is when they can predict the type of gene, the genes this particular sequence code for. So when they actually gives you this particular genes that this nucleotide code for, then there are you can copy these genes and also take it to another database to check their variance factor, right? To check the function of this gene. What are the function of this gene? So when you get the function of this gene, that's when you will know, okay, this gene is actually responsible for the pathogen of this particular organism. Now let me explain better using our research work. Now there is this particular gene. There are a lot of genes in these sequences or in this sequence. Let me say that. So there are a lot of genes there present. So some of these genes, for example, for this EPEC that I was talking about, some genes are just responsible for the attachment of the organism to the host, right? So when this gene is just responsible for the protein that will actually make this organism attached to the host. Now, so another gene is responsible for allowing this organism, after attaching, to penetrate in into the organ where it is being attached, right? So another gene is responsible for releasing the pathogen, that is the toxic substance of that particular organism, into the host. So the genes actually have different functions. So for you to know the different functions of this particular organism, so that is where this specific database comes in, to be able to detect the genes, predict the genes and their functions. I hope I answered your question. So mere downloading the DNA sequence, you can't actually predict if this particular um, organism will cause what, will cause defect or disease. Do you get what I'm saying? Now, a perfect example is Drosophila. Now, Drosophila is a vector-free organism. That is, Drosophila is harmless, cannot transmit uh, disease or vector or pathogen to any organism. So that's one of the advantages of Drosophila. So Drosophila is a vector-free organism that does not transmit diseases, unlike mosquito that can transmit what they call malaria, that can pass malaria from one person to another. I hope I've been able to answer that question. So, more than only the DNA sequence, you can actually know the pathogenicity or valence factor of that particular sequence until you subject it to another specific database.
All
Thank
right.
you very much.
Thank you, Ma.
Uh,
I believe Toyin Juliana must have been able to achieve the goal of asking that question. So thank you, Manuel. If there is any
yeah,
other
so
question, okay, you want to say something?
uh, yes, I want to appreciate that means you really followed
Hello,
the
Manuel.
conversation. Yes, I'm actually saying something. I said I want to commend her. That means she followed me through the presentation. Are you still there, sir? Network is bad. So I hope, Juliana, if I've answered your question, you can just type it in. Okay, thank you. Thank you so much. Thank you so much. I really appreciate you. Thank you for the patience. You can always reach out to Anatomy Triad for more information. All right, thank you so much. Do you have any other question? Right, so I don't know what's actually wrong with our chairman network over there, but I think we really have a wonderful session. There are quite a lot of things we can actually learn, but I desire that this is actually in series so that we can do some hands-on training.
Okay, Mr. Manuel, can you still hear me?
Yes, sir. Very much, sir. You're welcome. Hello, can you hear me, sir? Okay. We lost him again, but as well. Okay, is this what they call dry lab or is another thing? That's my last question. All right, so this is not a, yeah, this is a dry lab. Yeah, this is a dry lab. When you say dry lab, it just means I'm not working with um I'm not working with what's it called, animals, or you're not performing any physical experiment. Everything is based on computer, right? Computerize everything. You carry out your research using your system, algorithm, you know, just through database. So this is what we call dry lab. While when it involves you um dissecting, making use of organism, different organism, whether culturing or anything, that is what we call wet lab. So this is a dry lab, doesn't require any physical experiment.
All right. Mr. Manuel, I believe you've been able to answer his second question, or if that is what they call dry lab.
Yes, sir. Yes, sir. I've answered that. I just finished answering it. So I'm expecting a comment to confirm if she got my message.
All right. So, Tony Juliana, thank you for your continuous asking of question. We really admire your zeal to learn from bioinformatics. All right. So when you just confirm that, that's okay. We will see it on our chat via YouTube channel. So if there is no other questions from any other person, Mr. Emmanuel, on behalf of the directors, board of directors,
You can't hear me. You can't hear me. Board of directors. All right. If I can hear you, maybe it's network again. But I think it's trying to appreciate me, and I think I'm the one to show more appreciation to actually come to a platform like this is something I will never take for granted. Thank you so much for the invitation. Thank you, everybody that listened from the beginning to the end. I can't but thank you big, right? So this means a lot. Let's strive our best to make sure we we do the right thing. We have to do the hard work. Just let us just learn, continue to learn, study. It wasn't easy for me as an anatomy student. This is not about origin and insertion of a particular muscle or the gross anatomy of anything. It's not even histology, right? So this is more of cellular and molecular. So, but I was able to dive in, and it was quite interesting. It's actually quite interesting. So let's keep it up. Let's keep the hard work. Let's do the hard work.
All right.
You're welcome, sir. You're welcome, sir. Yes, sir, can you hear me?
All right. Emmanuel, can you hear me?
Yes, sir, very much. I can hear you, sir. So anybody listening to me right now, if you still have a question, you can actually ask. Always ask questions. Important. That's the only way to get knowledge, to have more insight or something. All right, so I'm waiting for you to just come up and then, okay.
All right. Emmanuel, can you see me now?
Yes, I can see you and I can hear you, sir.
All right. It's very bad over here. Like network is minus 10, not even plus one. So
I
I'm
understand,
using
sir.
different system to connect different access to internet, yet it's misbehaving. So I want to say a very big thank you to everybody that participated. Tony Juliana, thank you, and also our HOD from Prince Abubakar Audu University, Kogi State. We realized one thing: your passion to always learn and to transform young people in the biomedical setting isn't a small one. So I wish other HODs in the biomedical field in Nigerians that are currently watching or will eventually watch this, what we are doing, will be able to key into this mindset in which you have in transforming young people. You put in all your best to make sure that young people in biomedical setting not only just go through universities, but have all it takes to become problem solvers and to better their future. We celebrate you, sir, and we are very, very grateful for always partnering with us via the Anatomy Trial Foundation to talk to your students and to go there, not only physical, as they call virtually, but live. We are grateful, sir. We are grateful, sir. All right. So thank you, Mr. Manuel. Also, I noticed that Lautech is doing very great. Lautech is doing very great. It's exposing our young people in biomedical setting into various fields, not just theoretical base, but hands-on training. Even the doctor who is in charge of your Eagles lab,
Yes,
we
sir.
really, yes, we really appreciate her. We are seeing how she's making a move. Also, if you go to Ondo State too, we are having associate professor, if not really a professor now, Emamus is also currently doing very great, training young people in the field of biomedical sciences. We are noticing what you people are doing, all the areas of anatomy, physiology, biochemistry, pharmacology, and other field of the paramedical, basic medical sciences, how they are contributing immensely to grow young minds into becoming problem solvers. We are very much grateful. So here in Anatomy Trial Foundation, that is what we do for a living. We are not working for money here. All what we are doing is we want to make a difference. Breeding young people who will come up to not just study a course that is theoretically based, but a course in which they will be able to use to solve problems and earn money for themselves and turn away from poverty to becoming men of great influence, not just only in Nigeria and in Africa, but globally. So Anatomy Trial Foundation is covering not just only students, it also covers graduates, it also covers people who work in various industries, so as to bring all of them together to see it to reshaping what we currently call education as it concerns biomedical sciences or basic medical sciences. So if you have any question, you have any contribution, you want to sponsor us, we normally sponsor students for their grants in research writing, even during their undergraduate projects. We normally give scholarship to students also to aid them in studying their programs, and also we still need to organize some fundraising for students who also have opportunity to go for workshops and training that will enable them to have all the skills needed to do very well in biomedical sciences. Without biomedical sciences or basic medical sciences, there will be no better research in Nigeria or Africa, and there will be no better
Mm.
outcome in the so we'll be eager to do more and bringing more persons, even live sections and hands-on training. Thank you and bye-bye from our end. All of us in Anatomy Trial Foundation loves you all and God bless you.
Thank you so much, sir. Thank you so much. I really appreciate everything. Thank you. Thank you.
To learn to teach others. We are very, very grateful. Thank you very much. Thank you very much, Emmanuel. Thank you very much. Thank you very much. You have done noble. So let's keep our eyes open to see that very soon we'll kick up the introduction to bioinformatics. And if you are interested in learning bioinformatics, please do well to reach out to us. We can connect you with Emmanuel, and also Emmanuel can link you to the various hub in which he's learning bioinformatics that made him to be this very good. So if you're interested, link up to us and then we'll do that. So thank you on behalf of Anatomy Trial Foundation. We say thank you very much. Subscribe to our channel so that we'll know that.
All right. So thank you very much for having me there. And such a privilege, actually. Thank you. Thank you. I